Hello. Feel free to say hi. Once we get started, we'll get people to try and mute it. I'll put a message in but if you've got questions, feel free to raise your hand. I'm sure you guys have worked out how to make this work.
Get to the chat. Nice.
All right. I'll be right back. I got to answer something.
Okay.
Oh, holy good Lord. Okay. I'm trying to like arrange my screen so that I can see everything. I'm not sure how good that's working.
Hey, Venus here.
Hey, nice.
That's what I was gonna do.
Yeah.
I wonder if this works. I've been told.
Wow. How do I make it so that I can see raising hands while screen sharing? Okay. It's 2:30. I think we'll give it maybe another minute.
Hi. Have we started yet? Or?
No,
We have not, but I
still.
think we might start now. All right, so
Yeah, I've got to attend to something for a sec, so I'll just let you do it.
Cool beans.
Right.
Hi everyone. Thank you for coming to our first session. We know this isn't ideal, but I think we're all trying to work with what is available. Today I'm going to be taking you guys through cells, so everything that's in a cell and how cells proliferate and how they die, basically. I'll try to get through this as quickly as possible so that Subkan can take you through body fluids. This is a Zoom session, so I'm sure you guys are pretty used to like working with that. Please have your mic unmuted and raise your hand if you have a question or send it through to the chat. Yeah, so Subkan, if anyone raises their hands, please let me know so that I can answer their question. And towards the end of the session, we'll ask you to jump over to the next session or the next Zoom meeting that we've set up so that we can keep going, basically because Zoom only lets us do this for 40 minutes at a time. So with all that done, let's get started. All right, so cells. Nice. So prokaryotes and eukaryotes. Basically, to compare them, prokaryotic cells are older, simpler in internal structure because they don't have membrane-bound organelles. They divide by binary fission instead of mitosis, like we'll learn later for eukaryotic cells. And their genetic material isn't bound by a nucleus, they're just sort of dispersed randomly throughout the cell. So within eukaryotes, which we'll be focusing on for the first half of semester 1, there's a few important components. The plasma membrane, the nucleus, some organelles. I think mitochondria and ribosomes are probably the most important for now, and the cytoskeleton, which is probably like less important. All right, I think it's important to know the distinction between these two terms. So the cytosol is the fluid within the cell and everything that's dissolved in it. So all the proteins, all the gases, the cytoskeleton included in that, that's called the cytosol. The cytoplasm is the cytosol plus everything else that's in the cell except the nucleus. So one way you can think about it is the cytoplasm is everything that's within the cell membrane, but outside of the nucleus. Yeah, so let's move. So the plasma membrane, basically the purpose of the plasma membrane is to control what comes in and out of the cell. And the way it does this is related to its structure. So it's made up of 2 layers of phospholipid molecules, which form a layer that allows the membrane to be selectively permeable. So different particles have to move differently through the membrane, and that's due to the structure of the bilayer, basically. There's cholesterol in it, which maintains its flexibility. There are proteins, which allow some things to go into the membrane if they can't diffuse through naturally. And there are carbohydrate chains. All right. And there are carbohydrate chains that help the cell to express its identity, which is important in cases such as the immune system, for example. So moving on to the nucleus, it's basically made up of 2 layers of membrane with pores in the layers. The pores allow enzymes to come in and out of the nucleus to interact with DNA so that you can either produce proteins or you can replicate DNA for mitosis or meiosis. There is genetic material inside, which is just kind of important. And there is also nucleoli, which are composed of RNA, and they produce ribosomal RNA, which makes up ribosomes for the rest of the cell, and those make proteins, which are also important. So for the organelles, mitochondria, powerhouse of the cell, pretty important. They're made up of a double membrane. The outer membrane is basically just a sac. The internal membrane is more folded, and within that space on the inside of the internal membrane is a space called the matrix. Within the matrix is mitochondrial DNA, which is important because it codes for proteins that are involved in energy production reactions, which produce energy for the rest of the cell to use. There's also ribosomes, which helps to produce these proteins. And the mitochondrial DNA is also important because it is only inherited through the maternal line. So you only inherit your mitochondria and therefore your mitochondrial DNA through your mother. And this can help in tracing genes back through history, basically. There is a theory called the endosymbiosis theory, which hypothesizes that mitochondria were basically once bacteria that were taken into a cell and have stayed there for the rest of time. So this is a diagram that illustrates that you can look at later. This drive will be on our drive, so you can have a look at that later. Yep.
The link is in our description.
Yeah. So ribosomes are not membrane bound, so it's important to remember that these are the only organelle that is present in both prokaryote cells and eukaryotic cells. Ribosomes are basically there to produce proteins for the cell to use. They're made up of two subunits of ribosomal RNA, a large subunit and a small subunit. In between, there's a strand of mRNA, which is basically a messenger, a derivative from DNA that helps to produce proteins. And there is also transport DNA, which brings amino acids over from other places in the cell so that the ribosome can produce proteins. And you'll learn about that more later when you learn about central dogma. Rough ER and smooth ER, there's a few differences. So smooth ER is more folded like tubes, whereas rough ER is folded more like sacs. And rough ER extends straight out from the nuclear membrane, whereas smooth ER extends from the rough ER itself. Smooth ER is to do with processing lipids, whereas rough ER is processing proteins that are meant to be for secretion. So they're meant to be used outside of the cell. So the ribosomes are going to produce proteins which are taken into rough ER, processed and changed slightly, and then those proteins are going to be sent from within vesicles to the Golgi complex. So the Golgi complex is again just basically flat sacs of membrane that lie in parallel with each other. There's a cis phase which receives vesicles from the rough ER, and a trans phase which sends vesicles to the cell membrane. The Golgi apparatus, basically transforms and processes compounds that it receives, and then sends them out to the plasma membrane to be sent outside of the cell. This is a summary of how it works, and you can see them in the GIFs or the GIFs on the screen. Hopefully they're working.
Oh, wow.
So
yeah, they're working.
Yeah, and this as well, which is a little bit larger. So hopefully this helps you sort of visualize how the Golgi complex works. So as the vesicles are moving through the layers in the Golgi complex, they are changed slightly at each station, basically. All right, peroxisomes and lysosomes are there to break down stuff in the cell that the cell doesn't really want. So peroxisomes break down lipids, alcohol, and some components of the myelin sheaths, which are basically lipids. The one thing to note is that as they're breaking things down, they produce hydrogen peroxide, which is a toxic byproduct. So to nullify that, peroxisomes also contain the enzyme catalase, which breaks hydrogen peroxide down into water and oxygen. Lysosomes are there to break down cellular material that isn't wanted anymore. That can be things that the cell has ingested from outside of the cell, or just stuff inside the cell that wants to be recycled. Yeah, here are some micrographs of the cell and a diagram. And this is showing the difference between phagocytosis and autophagy. All right, the cytoskeleton. There are three main components of the cytoskeleton. There are microtubules, microfilaments, and intermediate filaments. So we'll start with microtubules. They are made up of these components called tubulin dimers. These tubulin dimers sort of join up to form a spiral that then forms a long fiber like this here. And the fiber extends beyond the end of the picture, basically. So it has many, many functions. Microtubules firstly maintain cell shape by weaving throughout the cytoplasm, as you can see in the green in the micrograph here, and it maintains the shape of the cell so that the cell isn't flopping all over the place. They are also involved in movement. So cilia and flagella are small extensions of the cell membrane into the extracellular space. And because it's membrane, it's a little bit floppy, it wants some structure. So basically what the microtubules do is give it some structure by also extending a small section into this extension of the cell membrane, basically. And it does this in a 9 and 2 arrangement. So each tubulin microtubule fiber will form up or join up with another one to form a dimer, which you can see over here. And there will be a ring of 9 dimers on the outside and then 2 microtubule fibers on the inside. So you can see that there's a 9 and 2 arrangement. And these cilium fibers then will provide structure to the cilium and the flagella, sort of like fingers in a glove. So if you think of your fingers as the cilium microtubule fiber and the glove itself as the cell membrane, the fingers are giving the glove some structure, and it helps the glove to move or bend in the way that the fingers want, basically. So, yeah. The microtubules are also important in mitosis because they form centrioles, which are 9 triplets of microtubule fibers. They are responsible for separating the chromosomes in mitosis and meiosis. So if there's something wrong with the centrioles, there can be problems with cell division and cell replication. Microfilaments, there are 2, actin and myosin, and these will come in they will be really important when you learn about skeletal muscle cells because they facilitate muscle cell contraction, which allows your body to move. They also maintain the shape of cells and they move organelles within the cell. They also aid in cytokinesis in cell division, so that's when the cell separates into 2 daughter cells. And intermediate filaments, all we were really given or taught about it is that they're a family of filaments. We weren't really told anything else, and they didn't come up again later, so apparently not very important. All right, so that's it for cellular biology. We're going to move on to cell proliferation and death. All right, so the cell cycle and mitosis is to make more cells, meiosis is to make gametes or sex cells, and apoptosis is to eliminate some cells that are not wanted in an organized way. So we'll start with the cell cycle and mitosis. So the point of mitosis is to proliferate, and this is to either promote growth or promote repair. So you either increase the number of cells within the organism or you replace cells that have been damaged. And the end goal of mitosis is to produce 2 daughter cells from 1 precursor cell. Basically, this occurs through doubling the number of DNA and the number of organelles, and then physically dividing into 2. So I already made that joke.
I didn't hear it. Big sad.
No, on the slide. All right, so in the cell cycle, there is interphase, which includes GAP1 or G1 synthesis, and GAP2 or G2. There's also G0 or G0, which I'll talk about as well. From G2, the cell moves on to mitosis, which includes prophase, metaphase, anaphase, telophase, and cytokinesis. Not telekinesis, as I accidentally typed when I was making this slide. All right, so G1. So G1, basically what occurs in G1 is the cell is growing. It's replicating everything in the cytoplasm, so all the organelles and all the proteins is being doubled. And because of this, the cell is doubling in size. The point of this phase is just to grow to a large enough size where the cell can survive, or it can keep going and get even bigger so that it can divide. So at the end of G1 phase, the cell has 2 options. It can either enter G0. So G0 is a resting phase where either the cell doesn't really do anything or it differentiates into a more specialized cell instead of dividing further. And what it does will be prompted by cellular signals. Some cells will never leave G1 and they will specialize or differentiate into a more specialized cell. But also some cells will never enter G1. They'll just keep dividing and producing more and more cells. And this is really important, for example, in skin cells, because you don't really want to run out of skin cells. You want to keep making those cells. So that's one option. The other option is to enter synthesis phase, which will lead to cell division. All right, so before the cell can enter division, oh, it says.
Oh. Okay, that's interesting.
Oh, okay.
Cool.
Okay. Well, that's good.
So I guess
Yeah.
we don't have to. All right, so
Time to
we've
switch
just
over.
received an announcement that we don't have to move on to another
I'll fix
meeting,
that.
which is great.
I think everyone saw that. Yeah.
Cool.
Yeah, but
Nice
like
job.
people, if anyone wants to join late, you know.
But while we're
That's true.
on a break, does anyone have any questions? Have we been keeping up all right?
If you have any
Oh,
questions,
right.
can you send it through to the chat?
Wait, Khan, are you recording this?
I am recording this, I'm fairly sure.
Yeah, okay. So,
Yeah. If you've got any questions, just raise your hand anytime. Any one of us will pick up on it. What's an example of a cell that never goes on to divide?
I'll be back in a moment.
So as I said,
I'm pretty sure nerve cells always stay G0 from my knowledge. So nerve cells actually never divide or replicate. So once you lose a nerve cell, we can't replicate it. That's why people get paralyzed. So essentially, I would say nerve cells. Yeah.
So that's one example. Yeah. Cool. All right. So anyone else?
Okay, another question
I
we've
was
got
just
from,
about to move on to cyclins and CDK, so I'm going to get to that in a little bit. Anything else anyone wanted to add? All right, so I'm going to keep going. All right, so before the cell moves on to replication of the DNA, it wants to make sure that nothing's gone wrong in replicating the cytoplasm. So it basically does this through two genes, the retinoblastoma gene, so the RB gene, and the P53 gene. Basically, what both of these do is they are active during G1, and while they're active, they do two things. They check for faults within the cell, and if there are faults, they are inactivating genes that allow the cell to move on to S phase. So because they've blocked those genes, when they are active, the cell cannot move on to S phase. So if there is something wrong, then the cell can't keep going into the next phase. But if there is nothing wrong, or if something has been fixed, these genes will be inactivated. And so the genes that allow the cell to transition into the next phase are activated, they can be expressed, and so the cell can move on. If these genes are defective, if they're not functional, the cell cycle can't be controlled. The cell will be replicating with faulty organelles and faulty genes, and that can lead to cancer, basically. But if they are active and the cell can move on to the next phase, some genes will be expressed, and these are cyclins and CDKs. There are a family of proteins that regulate the transitioning of the cell from one phase of the cell cycle to the next phase, basically. And all you really need to know about it is that each phase of the cell cycle will have one cyclin and one CDK that are active mainly in that phase. So each cyclin will have one CDK paired with it, and the function of these pairs will basically be resulting in production of another cyclin, which then prompts the cell to move on to the next phase. So we've covered gap 1. So in gap 1, the cell currently has 2 copies of each chromosome. So there will be 2 copies of chromosome 1, 2 copies of chromosome 2, so on and so forth. In S phase, the DNA is replicated so that there is 4 copies of each gene, but still only 2 copies of each chromosome, if that makes sense. So there will be 2 copies of chromosome 1, but each copy will have 2 chromatids, so that in total there are 4 copies of each gene. I hope that makes sense, everyone. But
You got
hopefully,
the two. You got the two. You got two of these.
two
Yeah,
of these.
pretty much.
Two of these.
Yeah.
Yeah. So hopefully there are some diagrams that will help you visualize that as well. So basically in S phase, the DNA is replicated. It's moving on to the G phase, where the cell is making sure that daughter cells can survive, and again checking for mistakes in the DNA. If there are mistakes, it's going to reactivate p53, which will inhibit moving on to the next phase, which is mitosis. But if there's nothing wrong, or if all the stuff that's wrong has been fixed, then p53 is inactivated again, and the cell can then move on to the next phase, which is mitosis. So in mitosis, there are 4 parts. In prophase, the DNA is going to condense, forming some chromosomes that you can see. And each chromosome is going to have 2 chromatids, which are connected at a centromere. The nuclear membrane is going to dissolve, and centrioles are moving to either side or opposing sides of the cell and forming a spindle. In metaphase, spindle fibers are growing and attaching to centromeres. In anaphase, the spindle fibers are contracting, and that will break the centromere and separate the chromatids. So now there are 4 copies of each chromosome, and the cell will distribute these copies so that each daughter cell has 2 copies of each chromosome. In telophase, the nuclear envelope reappears. There's a furrow which starts to pinch the precursor cell into 2 cells, and cytokinesis will basically separate the one cell into 2 daughter cells, which will be identical to the first cell that we started with. And then these daughter cells are going to re-enter interphase. They are going back into gap 1 and then either into G0 or into S phase again. And then the cycle keeps going. So that's some more, lots of diagrams.
Anyone have any questions about that, or just before we move on to the really complicated meiosis bit? Yeah, we all good? Nothing? Okay.
Cool. So meiosis, we're doing meiosis to make gametes or sex cells. So eggs in females and sperm cells in males. And this diagram up here sort of illustrates the difference. You can see that in making egg cells, it results in one sort of big egg with 3 polar bodies. And this is because there's a lot of nutrients that are necessary for the survival of the embryo and the development. And so all the nutrients from these polar bodies are given to the one egg. Whereas in making sperm cells, you just make 4 equal sperm cells. So yeah. So in meiosis, basically it's mitosis, but twice. You have prophase, where the chromosomes are condensing, but more importantly, homologous chromosomes are aligning and crossing over. So homologous chromosomes are what I said before. If you have 2 copies of chromosome 1, those 2 copies are homologous chromosomes. So in prophase 1, they are lying right next to each other and they are crossing over so that recombination occurs. And I'm going to skip ahead to this. All right, so as you can see here, these are homologous chromosomes. They have the same genes, but they have different alleles, right? So alleles are basically slightly different versions of each gene. They will produce a different sort of version of the trait that manifests in your body. So basically you can have a gene for eye color, for example, and one allele might code for brown eyes and one allele might code for blue eyes. So that's the common example that people use. So in prophase 1, the homologous chromosomes are going to lie next to each other. They're going to cross over and they're going to swap alleles, which is called recombination. And here's a more complex example over here where you can see there are lots of different combinations of chromosomes that can occur from these 4 sort of copies of the chromosome. So if we go back to here, in metaphase, the metaphase and anaphase 1, the homologous chromosomes are separated, but the chromatids are not broken yet. So in telophase, each homologous chromosome is separated into sort of one side of the cell. In prophase 2, nothing really happens. In metaphase and anaphase 2, the chromatids are broken, and so the sister chromatids are separated. So that results in one copy or one chromatid in each daughter cell. And that's important because you want each daughter cell to have one copy of the genetic material. So rather than being 2N, like normal somatic cells, you're going to have 1N, so one copy of each chromosome. Yeah. There's some stuff here about ensuring genetic diversity. So crossing over and recombination is one way to ensure that. And there's also independent assortment, which is basically how each chromosome is, or how do I explain this? Basically how different combinations of alleles and genes can be sorted into each daughter cell. Want to get quickly through this, so have a look more closely at the slides later. But basically it ensures that daughter cells are different from the precursor cells so that you're not identical to your parents. And this is good because within a population, it allows genetic variation, which can contribute to the survival of the population and the species. There are some bad stuff that can occur during... Hi, Nikita, did you raise a hand?
Yes,
Nikita's
I
posted
did.
a table.
I just quickly wanted to say something. So in terms of that genetic variation, there's 3 ways. So you mentioned the crossing over and independent assortment. Yep. There's also one more, guys, and that's called random fertilization. So if you think about it, in terms of ovum, so the eggs and sperm, each of them have 8 million different sperm and eggs, right? So there's basically 64 trillion possible diploid combinations. So that's the 3rd way. Just, yeah.
Yeah. So the end result is that the end result is that there's a lot of different combination of alleles that can occur. All right. So complications of meiosis. Sometimes the centromeres don't separate properly, so you can get non-disjunction, and that leads to cells with more copies or fewer copies of a chromosome than normal. And you'll learn more about this in population genetics as well. So aneuploidy is like an abnormal number of chromosomes in a cell. Complications in spermatogenesis. So sometimes the sperm cells are attacked by immune cells. Usually this doesn't happen because of a blood barrier between your testes, where your sperm cells are being made, and the veins. Sometimes that blood barrier doesn't occur, and so the sperm cells are attacked by immune cells, which are leading to infertility. Can also be caused by other conditions, but this is one way it can occur. Cell death, we're nearly there.
Yay. Okay.
Apoptosis and necrosis. Apoptosis is much more planned. It's regulated and started by the cell or by the body itself, whereas necrosis is due to external factors. And it causes inflammation. There's a lot of debris, which if there is a lot of dead build-up tissue, then it needs to be surgically removed, or it's going to damage and affect the rest of the organ, or maybe even the whole body. So it's important to have apoptosis in some, in some cases, making sure that organs are sculpted properly and so that they can perform their function. Apoptosis can be initiated in 2 ways. The extracellular way, extrinsic pathway, is when there's a signal from outside of the cell. It will be received by receptors on the cell membrane, which then activates a caspase. So caspases are a family of proteins that are responsible for apoptosis, basically. There's many of them, from caspase 1 to 9 or 10. In the extrinsic pathway, caspase 8 is activated first, and it activates 3, 6, and 7, which are the executioners, basically. In the intrinsic pathway, the signals come from inside the cell, and in this pathway, cytochrome C is released from the mitochondria, which activates caspase 9 first, and caspase 9 then activates 3, 6, 7, which again executes the cell. So the only thing that's really different is which caspase is activated first and where the signal comes from. So there's the pathways illustrated for you guys. So the actual execution, the chromatin and DNA is condensing, the nuclear membrane dissolves, and all the DNA becomes fragmented, so into little sections. The cells begin to bleb. So they're forming little sacs of cellular material, and these sacs are then phagocytosed by immune cells, which cleans up the cellular debris. It breaks down all of the cellular material within the blebs, and then reuses them or recycles them for other purposes. Somebody asked, do we need to know which specific caspases? I don't remember it being too important, but I
don't remember it coming up on a question.
Yeah, no. So I think just know that sort of the differences in how the extrinsic and intrinsic pathways are started. So where the signals come from, basically. So yeah. And I think that's it for the execution. I thought this image was really cool. You can see the cells start to bleb and form little pockets, little sacs. And here's some diagrams again.
Have you guys had this lecture yet on apoptosis and cell death?
They have, yeah, last week.
Yeah, okay, cool. All right.
I guess the only extra thing I'd add is just in terms of not being stressed about trying to know everything, but knowing how it fits into the bigger picture. Like all of this is going to be more important when we do other lectures, and it's just going to form the basis of that. So don't be too stressed if you don't know like specific numbers about like which caspase or which cyclin it is. That's not the point. The point is for you to know that actin and myosin now exist and that they're going to fit into muscle cells later.
Yeah.
So
even if you don't remember, it's good just to understand. So even if you don't remember, you can recall it later again. No one's going to kill you for it.
Yeah, so I think that's true. Like this is sort of forming a basis and a background for you guys to call on when you start doing more specific topics like central dogma and cancer, cancer, skeletal muscle cells, all of that. This is just the background knowledge that you need. And I'm sure a lot of you probably did bio or the bio bridging course. So this probably isn't too much new stuff, but if it is, don't stress too much about it. You know, there's always more time for you to revise this.
Yeah, and if you've got questions about anything that we've covered so far and that's coming up later, you can always shoot us a message and we'll be happy to respond.
So before we move on to body fluids, does anyone have any questions?
I'm going to take that as a no.
Yeah.
Just anytime you put up a hand, just we'll reach out and get that. Yeah.
Selkan, do you have the slide up, or do you want me to keep?
I do have the slide
sharing. All right, I'll make you host then. Stop share. Selkan, make host. There you go.
All right. So yes, I need to start sharing the video. That one. All right.
What is happening?
I don't know.
Oh wow, this is this is how it should usually look if you screen share. All right. Body fluids.
All right. Yeah. So body fluids. So I've heard that you guys actually, Chris Wright walked out of your lecture, which is Crap. Okay. I'll be back in a moment. I need to get a charger.
Wow.
Yeah,
That's
okay.
right.
So in reference to body fluids, like what I'll say is that there's essentially 4 different body fluids, and if you kind of learn how they affect the cells right, you're going to get the exam questions right. There's only 4 questions that they could possibly ask you on the exam about this, and everybody pretty much memorized it and got it right. So understand it, but don't be too worried. Like it's about 2 questions out of, I don't know, 90 questions on the exam in
It's
terms
not
of
much.
multiple choice, and it's pretty much just memorization. So yeah, and the clinical application relates to IV fluids, and he's back.
Yes, hi. Yeah, okay, so basically I've heard that Chris Wright walked out of your lecture because of technical issues, which would have been hilarious to see, but we didn't get that. So we actually got a full lecture. So body fluids, basically, he starts, like I'll try and cover what he went through, but it's probably not going to be as good, sadly. But that's okay. So pretty much the background for it is the human is 60% water, the human body. And 1/3 of that is found as extracellular fluid, and 2/3 of that is found as water within the cells as intracellular fluid. So because we have 60% of us as water, our concentration of water inside the body is like 55.5 molar, which is really, really high, especially compared to the next one, which is like sodium at 16 point something, I think. And what that means is water greatly influences how things work inside the body.
Stay hydrated.
So, yeah. So in terms of ions and membrane permeability, these come into play in the body as well. So ions, pretty much your potassium and phosphate ions are more abundant in the cell, and then sodium and chlorine and, oh man, I forgot what that was, HCO3. They're more present on the outside as well. That'll become more relevant later on, but for now, yeah, it's just useful information, I guess, later. Membranes, they can be permeable. So membranes in relation to, no, ions in relation to membranes can be permeable, effectively impermeable, and non-permeable. So basically they can either pass through, but then get yeeted out, or just not be able to pass through at all. So we have this little funny graphic here. Yeah, not sorry.
Turning into code.
So, yeah. Yeah, so next concept that they go through is diffusion. So basically it's the net movement of particles down the concentration gradient. Common definition, you might have heard it. Did someone, are you getting chat?
Yeah, it's Venu just mocking you. Don't worry about it.
Yes. I mean, yeah, okay, it might be pretty bad. Have fun, guys. Yeah, so so that's just like the classic definition that you get from like chem and bio. It's a little diagram you get here. The reason I didn't put the membrane in the middle, like the orange line is the membrane. Reason I didn't put that in the middle is because then you might get confused and think it diffuses like perfectly evenly, whereas it's not the case. So it's got to be distributed proportionately towards however much space it is. In terms of factors affecting the rate, it's pretty much the same. So temperature, weight of molecules, concentration gradient, membrane permeability, and membrane surface area.
And just to add upon that, diffusion is the movement of the solute, whereas osmosis
yes.
is the movement of the solvent. So don't get confused between that.
Yeah, I think next slide's osmosis anyway.
So often you'll say something diffuses across the membrane. That's a good way to put it, usually.
Yeah. So osmosis, that's the net movement of water molecules up the concentration gradient. So the concentration gradient refers to the concentration of the solutes. So in terms of, if you put it in terms of the solvent, it would still be moving down the concentration gradient. Just fun facts, I guess. Yeah, So that's that. And then osmotic pressure is the amount of pressure that you need to stop the movement of water. Yeah, so I guess if osmosis is this entire diagram on the slide, then your osmotic pressure would be the amount of pressure you need to keep it in that initial state.
And it'll be pushing down on the left side.
Yes. And then in terms of how you determine that, since pressure is force over area, it depends on the amount of collisions. Basically, it's determined by the number of particles involved. So it's what's called a colligative property. But then you're basically never going to hear that word again. So just know that it's determined by the number of particle rather than the nature. Osmolarity and tonicity. Osmolarity is the number of solutes per liter, and it takes everything together, as opposed to concentration, which would be just the amount of that one specific solute. So if you had, actually, yeah, no, there is a question next, but yeah, an analogy that the PSP that we had for our year level, or one of them at least, taught us was that you have osmoles, which are not racist, and you have concentration, which is racist. So basically, you take everything together and you get osmoles, and then if you take things one by one, then it's concentration. So I've got a quick little question. I'll get you guys to drop your answers in chat. You have a solution filled with the molecule AB at equilibrium. What's the osmolarity of the solution? And then dissociation constant is 0.8, which just means that 80% of the molecules in that solution will dissociate into A plus B. If anyone's feeling up to it, I'll give you guys about 30 seconds or so.
Even if you're right or wrong, it's a matter of just put it out there. Be brave.
Be brave and share.
And don't worry if you're wrong, because that just gives us a chance to like discuss.
If you're wrong, all right.
Nikita doesn't even know the answer.
That's great, Nikita.
Okay, someone's gone with 1,800 osmoles. Yep. Wait, milliosmoles.
Yeah. So just 1,000. Yeah, that's right. So basically, for anybody who's wondering how we got to that answer, so we have a. Wait, did I? Yeah. Okay. So to work out how we got to that answer, I'll just change what. I want to try something.
You can write it on using the annotate if you want.
Where's annotate? Oh,
you got to view options at the top. Yeah.
Got it. Basically, what happens is you get so dissociation constant is 0.8. Oh my god! Right. Let me just.
You can type it as well if you like.
I refuse to type. Oh my god, that did not work. Okay. I usually have a button which lets me erase things, so that did not work. Big sad. All right, so you have 0.8. Where's the control?
We love Zoom, guys.
Sorry, it's my first time using this. First time user. Okay, so you have 0.8. So 0.8, so that just means that 80% of this molecule AB will become A plus B. So in terms of osmoles and molarity, that just means that you're going to have, oh yeah, one thing I forgot to mention actually, that might have helped. Osmoles is equal to moles. So it's just, osmoles are the sum of all the moles, I guess. Yeah. So what that means is if you had, let's give this an arbitrary, so you take 0.8 and you multiply that by 1 because there's only one molecule of AB, or 1 mole, I guess.
Probably should have said 1 mole of AB.
Yeah, I know. So because it's an equilibrium, so there's going to be, and there's only one molecule on that side of the equilibrium, it's going to be 0.8 times that. So in this case, we'll just take it to be, because it's the sum of all the different types of molecules on that side of the equation, So there's only one. So it's going to be 0.8 times 1, which is 0.8. And then on the other side of the equation, you have, oh, wait, no, sorry. That should be times 2. I've taken the wrong side.
0.2, yeah.
Yeah. So it should be times 2, my bad. Yeah, because we're taking this side of the equation. Because it's the dissociation constant, so that's how much it'll split. So then you add that to the rest of everything that didn't get dissociated, so it's 0.2, because 1 minus 0.8 is 0.2.
You got to change your answer to 1.6.
Right, yes, I do need to do that.
But you get the picture, everyone.
Yeah,
right.
That's 1.6, and that becomes times 0.2 times 1, so 0.2.
Okay, so we've got a question.
So there's a question,
Yeah.
Does volume ever matter when calculating osmolarity? It works like concentration. So it's given as a like osmoles per liter. So I don't think it does.
No, yeah, it doesn't matter. Because it's like saying given, moles of a thing is not
how much like you have per unit volume.
Osmoles so is the same. It's an amount. Volume. Yeah.
Yeah.
So really what they're trying to say, I think, is when you're looking at concentration, right, it's referring to the concentration of a specific ion. So if you're looking at the concentration of, let's say, chlorine, that's just 150 millimoles per liter. But when you're looking at osmolarity, it's referring to all the ions in the solution. So let's say we've got chlorine and we've got sodium in the solution, then, and both of them have a concentration of 154 millimoles per liter. The overall osmolarity is 154 times 2 per liter. So osmolarity doesn't discriminate between, I guess, what the ion is, whereas concentration really refers to something specific.
Another thing to add to that is a good way to remember is that when you talk about concentration, right, you are talking about molar, whereas osmoles, you can think of it this way: osmoles are moles, but they're everything, right? Because they have the same word inside them. So those are a discrete amount as opposed to relying on the volume. Does that answer your question?
I think when you're calculating osmolarity, because it does take into consideration like concentration,
if you want to say,
it would still consider volume, right? Am I wrong?
Oh, sorry. It depends on the word that you use. So if it's osmoles, so here it's how many osmoles, but if it's the osmolarity, yes, it will depend on the volume. So it's really easy to mix it up. So osmolarity is the same as molarity. Osmoles is the same as the same as moles. So here, if you actually want to find out what it's actually asking is how many osmoles are in the solution. But if you want to say osmolarity, then you would need a volume. Sorry for the confusing mix up there.
Yeah, it's a bit all over the place. I don't know. I never really, okay. Moving on, osmolality is basically the same thing, but with an L in terms of word. And that takes into consideration mass instead of volume. Pretty simple, straightforward. Tonicity is more relevant towards like the clinical side of things. Basically, it takes into account the number of non-diffusibles per liter. So like the ions from before, that's where those start to come into play in terms of cells and how they act in the body and the red blood cells. So that tonicity does incorporate both non-permeable solutes as well as those which are effectively impermeable. We got stuff in chat.
That's just me. Yeah.
Okay. Yeah. I'll keep that.
Oh, you want to clear the?
Yes, I need to do that. I don't know. I thought Zoom was. Oh no. Okay.
You got to go to the annotate tab and there's a clear.
Yeah, I know.
So while he's clearing that, I guess I'll just, so we don't waste time. In terms of hypertonic, isotonic, a smart way that my biology teacher taught me to remember everything was if you're thinking about hypertonic. The reason it's low in solute is because, first of all, we need to remember things move from a high solute concentration to a low solute concentration. So if you're comparing your bloodstream or pure water to a red blood cell, let's say, the red blood cell obviously has a higher concentration. So in terms of hyper, you think of it as hipper. So you think of the red blood cell becoming fatter, essentially like a hipper, and lysing, so bursting. In terms of the way that I thought about hypertonic, was the cell is becoming really, really hyper, right? So it's getting rid of its solute concentration and releasing it into the pure water. And then isotonic is just equal. So that's a way that might help you guys.
Still learning. Okay, yeah. Yeah, so osmolarity and tonicity, they aren't exactly the same. They do consider similar things, but they're not quite the same. So consider that you have a tub with two solutions which are separated by a semi-impermeable membrane. On one side, you have a solution with the osmolarity of 500 milliosmoles per liter. On the other side, you have a solution with the osmolarity of 500 milliosmoles per liter. Are they isotonic? Can I get a yes or no in chat?
Everyone just flood the chat. We don't care.
Don't all talk at once. All right, it's fine. I'm going to assume, all right, we're just going to move on then. Basically, they aren't isotonic, but they are isoosmolar. Basically, we don't know what's inside the solution, so we don't know if the solutes inside these solutions can diffuse across the membrane. So yeah, as Nikita was saying, this is where hypotonic, hypertonic, and isotonic come in. When you're describing it, you're describing things in terms of like the larger bath around it. So if you had a cell in a bath, you'd be describing the bath. So if you put a cell into a hypotonic solution, you're putting it into a bath, which would have the water diffuse into the cell because it'd try and equalize the concentrations. So
Is everyone following? Just to like put your hand up if you're not following, like if you don't understand what is going on at all.
Is anyone doing that?
No. Okay. Hopefully that just means that they actually meant that. They're not just like completely ignoring us. But
Also, I'm pretty sure, correct me if I'm wrong, but I'm pretty sure this is something that I used to get confused with. So in terms of red blood cells being put into solution, I'm pretty sure the whole point of that is that the solutes are impermeable in terms of the membrane. So it's osmosis, as in the water.
Yeah, so it's osmosis. Yeah.
And when the water is moving, it's moving from the low solute concentration to the high solute.
To the high, yeah.
Because if we increase the volume, the high solute concentration, we bring the concentration of that thing down. And that makes it closer to being isotonic, okay? Does that make sense? Is that okay?
So
please do raise your hand if that makes sense, actually, so we can see if you're following along.
Or just drop something in chat, so then we'll see it.
Yeah, okay, so we've got one hand up.
Yeah, so
I assume everyone else is... Yeah, we've got two. We got
a few. Okay. Will. You can either. Oh, okay. So
I'll say that again. So what I was saying in terms of putting a red blood cell into a pure water solution. Okay, the whole point is that the membrane of the red blood cell is impermeable to your solutes. Okay, so to make the concentrations equal, we have to rely on osmosis. So the movement of water. When we're looking at solutes, usually they move from an area of high solute concentration to an area of low solute concentration. But with osmosis, it's flipped around because it's the movement of water. So it's moving from a low solute concentration to a high solute concentration. And it's the water that's moving because the solutes cannot move to equalize that concentration, yeah? So if water goes inside a cell, what's happening is that it's bringing the concentration down by putting more water in, because we can't actually change the number of solutes on either side, yeah? Is that better? Okay.
Cool. Yeah, no worries. Yeah, all right, so we'll go back to there. Yeah, so ultimately that really just comes down to how osmosis works, pretty much. With osmosis and diffusion, basically what you'll have happen is the concentrations will begin to equalize. Oh, you can't see me. It'll begin to equalize, yeah, just as a result, because of how the concentration gradient works. Yeah, so clinically, red blood cells have a osmolarity of about 300 milliosmoles per liter. Numbers may vary.
Remember that number. It's one of the most important numbers. Like, memorize that number. You need to know it for everything. Just FYI.
It's like actually 298 or something, but just remember 300 and you'll be fine. Yeah.
Yeah.
It's important for this topic. So
it is. Yeah. So depending on what you use for the IV fluid, it will determine how the red blood cells fare. No, wait. Yeah.
Yeah.
So in terms of if you use a hypertonic IV fluid, you're going to cause crenation of the red blood cells. So they'll shrivel up because the water moves out of them, leaving, the water moves out of it to try and equalize the pressure in the bloodstream.
Just to be a bit more specific about that, in terms of hypertonic, so what you're thinking about is you're thinking about the water or that outside solution having a higher concentration, right? And the red blood cells having a lower concentration, okay? So the red blood cells are becoming hyper essentially, and the water is rushing out of them into the higher concentration, higher solute concentration solution side, right? Bringing that concentration down and bringing the red blood cell concentration up till they reach kind of like an equilibrium.
So with isotonic IV fluid as well, basically because the concentrations are equal, there's going to be no net water movement. So there will still be water movement, just it's not going to affect it too much because you have the same amount of water going in as you have going out. Hypotonic, so that means that the surrounding bloodstream is relatively low in, it's relatively low in ions. So the water rushes into the red blood cell in order to equalize the concentration, and then because so much water rushes in, it starts to like the red blood cell can't keep up. It doesn't have enough of the lipid bilayer to contain everything, so it bursts and goes and has hemolysis basically. It might help to think of yourself as the tub from before, as a tub with the red blood cells as little bubbles with semi-permeable membranes containing your solutes. What's next? So IV fluids, Chris likes to talk about 4 of them. So you have pure water, 5% dextrose.
So this is what you inject into a patient.
And 1.8% urea. The thing with the weird percentages is that's just what it takes to get it to be isotonic with your bloodstream. With pure water, that's the only one which isn't isotonic. It's actually isoosmolar, not isotonic. My bad.
These will be on the exam, as Nikita said. So what happens is that they'll give you a question. They'll say, okay, you are going to inject a patient with this much stuff, right? What do you inject them with? Yeah. Or what's going to happen? It's like they'll ask you, then we're injecting them with this, what's going to happen? Or they'll ask you, we want to inject something into them to give them more water, for say, or something like that. And you have to pick which is the right one. So it's like I'm trying to explain what happens if you do every single one.
Yeah.
Or
and
each
also,
one.
A common trick that they put in is that saline says 0.9%. They'll commonly write it down as 9% instead. So just to trick you. So
let's go through them. Let's go through them properly.
So pure water is hypotonic to red blood cells, and it because of that, it will cause hemolysis. So don't use this, even if they're like dehydrated or something. You can use one of the other ones for that. So 0.9% saline is isotonic to red blood cells, and as well as isoosmolar. And you can use that to increase the blood volume and blood pressure. So when the patient's lost a large amount of blood or something like that. I think it's also one of the more common IV fluids as well. So I think I vaguely remember someone telling me, just if you come across an exam question asking you what to put, just go with 0.9 saline.
Mhm.
So yeah, that's what I do based on that. But I'm not sure how trustworthy that source is. But hey, it's worked for me so far. 5% dextrose is initially isotonic, but when the sugar is used up, it becomes hypotonic. So quick
what
question,
happens? Yeah.
is that an issue? And then if no one wants to answer, which is fine, it's not an issue because what will happen is the dextrose inside, or the sugar inside the fluid, will be used up by the body. So because it's initially isotonic, it's not an issue. And then as the fluid starts to lose its dextrose, the body will adapt for the incoming levels of fluid and water. So it's not an issue. You can use it for dehydration, and it's pretty handy to have. 1.8% urea, don't use this. Like, don't use this. It will cause hemolysis. Or not, yeah, it will cause hemolysis because it's hypotonic and it's isoosmolar. So what will happen is the urea can also diffuse across the membrane, I think.
Yeah. So what happens is that the urea, if the urea will easily diffuse across the membrane, and that will actually increase the amount of ions in the actual cell, and then the water will still want to flood it.
Yeah, water will follow it to flood it, and then that'll cause hemolysis. So once again, don't use it.
Okay, so question is, what's the difference between isotonic and isoosmolar? So isoosmolar, go ahead.
So that comes back to this previous question here. Where did it go? There. Yeah. So you can have a solution. So that comes back to this question here, where you have a solution with a semi-permeable membrane in the middle, with two solutions of 500 milliosmoles per liter on either side. The only reason we can't tell, we know that these solutions are isoosmolar because it gives us that they're both 500, but we can't say if they're isotonic or not because we don't know what the solutes are. So if the solutes in the first solution are permeable for the membrane, but the solutes in the second one aren't, then they're not isotonic because you have a higher tonicity in the second one, because tonicity is about non-diffusibles and effectively non-diffusibles.
I think just to clear. Oh yeah.
Yeah. Sorry, Brendan. The difference between isoosmolar and isotonic, as I understand it, is that isoosmolar takes into account every single solute, like Subkan said. Tonicity only takes into account the things that are permeable, right? So
not permeable.
Or not permeable. Sorry, yeah. So tonicity
the ones
takes
that exert pressure.
Yeah, the ones that aren't permeable, and so they would cause movement of water across the membrane because they can't move across the membrane themselves. Yeah. So with this question here, you know that they are isoosmolar, but as Subkan said, you don't know which ones are permeable and which ones aren't. And so you can't tell if they are isotonic or not. Isotonic would mean they have equal concentrations of non-permeable solutes on both sides.
Yeah.
So I hope that helps.
Does that make sense?
Yeah.
All right.
Let's see. Okay. Yeah. So this is one of the questions that you can get. Oops, you guys saw the answer, but that's all right.
Are we going on time? Oh yeah. Okay.
Are we going on time? We're going. Okay.
It's like it's almost 1:40. So.
Yeah. We'll try it, or almost done anyway. So if someone's asked a question. So if their tonicity was the same, then nothing would happen as. Yeah. So if their tonicity was the same, basically nothing would happen. It'd be the same, basically. There's nothing special happens. You just won't have molecules crossing over the thing. You won't have molecules crossing over the membrane. Yeah, so you might get questions like this. How does the water move? So you have a bath containing, is that a question? Yeah, all right.
No, that's all right. We got it.
So yeah, you might get questions like this. So you have a bath containing a cell. In the bath is a solution which is isoosmolar with the cell, so 300 milliosmoles from before. And then you add a 1.8% urea solution. How will the water move? I already kind of shared the answer before. I didn't realize it, but basically, we'll just go through it, I guess then. Oops. So yeah, so in terms of the initial osmolarity, you have 300 inside the cell, 300 milliosmoles per liter inside the cell, 300 milliosmoles per liter in the blood stream. Then you add 300 milliosmoles of urea. So 1.8% urea is 300 milliosmoles as well. So the osmolarity inside the cell becomes 600 because urea is diffusible, so it can pass through the cell membrane. So it goes in and it equalizes that through diffusion. And then the change to the bloodstream is just taken as negligible, in case you're wondering what happens there. And then in the blood, it stays as 300.
What you have to understand is that we are assuming that it's an infinite bath. This is something that probably Chris Wright probably mentioned, but it's saying that basically because your red blood cells are so small compared to your bloodstream, whatever you change to your bloodstream is effectively not going to change the concentration within the bloodstream.
Yeah, because it's just that big, so it doesn't really make a difference there. So then you have a difference of 300 in terms of the tonicity. So you have 300 more, you have just 300 as a value inside the cell, and you have relatively 0 inside the blood. Then the water will want to make the concentrations of the solutes equal, so then it rushes into the cell, you get hemolysis, and then the red blood cell dies. So as a summary, osmolarity is the molecules per volume. It's also akin to moles as well, but it's just the sum of all of them. Tonicity is the non-diffusible molecules per volume. Water moves from low tonicity to high tonicity, but not necessarily from low osmolarity to high osmolarity. And that's again to do with just being diffusible or not. And water won't move if the solutions are isotonic, but can move if they are isoosmolar. Almost done. Homeostasis and acid and base. I think this is like one slide. Ooh, chat question.
Can you think of tonicity as the difference in osmolarities between the two containers? Well, the thing is we don't really want to mix up the actual terms. So tonicity is the difference in tonicity between, the difference in tonicity between two containers is what is the difference in terms of non-permeable solids. So if I have more non-permeable solids in one side, so let's say if I have a tonicity of 600 moles, 600 millimoles, something on the one side, and then 300 millimoles on the other side. There's a difference in tonicity of 300. Whereas if you are effective osmoles, so I'm actually a little bit,
talk?
Yeah, jump in.
I don't think it's effective to think of tonicity as difference in osmolarities, just because you can have something that's isoosmolar, so no difference in osmolarity, but still different in tonicity. If that makes sense. So it won't work in that case because there is no difference between osmolarity. So just think of them as two separate things. I think putting together might just confuse you.
Okay, I'll explain effective osmoles because that completely slipped my mind when I was making this. I'll explain that at the end, I guess. But we'll just go through over homeostasis and acid and bases because I think it's just one slide. It won't take too long. But basically, we didn't get a proper lecture on this. Professor Wright just spent about like half of it or something, maybe even more, on just clarifying the stuff that we've done up until now. So basically, just really quickly, normal body pH is about 6.8. That's not important. Don't memorize that number. It's a waste of time. Instead, memorize that normal blood pH is 7.4 plus or minus 0.05. That's the important one, because that's what you'll be measuring and keeping track of. And then in terms of deviations from 7.4 pH, you can get acidosis or alkalosis. I'm not sure if that's what it was. Might have made a typo there. But basically, your blood becomes either acidic or alkaline, depending on if it goes up or down 0.05 in terms of pH. And then if it goes up or down 0.6 pH, then it can lead to death, which is not good. That's what you want to avoid. Acidotic versus alkalotic. Yeah, I think that's probably better wording there. I'll change that on the slides for you guys as well. I think. Yeah,
any
so that's the last.
questions?
So any questions about that? If not, I'll quickly go through effective osmoles as well.
Yep. No. Okay.
Okay. So basically, I never really understood what effective osmoles are, to be honest, which doesn't sound great, but that just shows how high yield it is, which is not very, but it can help in terms of concept, right? So I'll just find something for it.
Nikita, do you want to try and explain that, or?
Just before that, I just want to go through some common questions that you could get on this, okay? So essentially, the first type of question is, what happens if you put a red blood cell in pure water, okay? So we know that the concentration of the red blood cell is high. Pure water is obviously nothing, no ions in it. The ions are impermeable, so they cannot leave the red blood cell. So what we have is osmosis. So it's a hypertonic solution because the concentration of the pure water is less than the red blood cell. So water rushes into it, causing lysis. Okay, so that's the first type of question you can get. The second question is what happens when a red blood cell is put into a bath of normal saline. So if you're looking at normal saline, essentially 0.9% is considered normal saline. I mentioned before how they trick this up with 9%, so just make sure you double check what you're reading. So if normal saline is isotonic to a red blood cell, there's going to be no movement of water, so nothing will happen. The next type of question is what happens when a red blood cell is put into a bath of isoosmolar urea. So I'm pretty sure this is the 1.8% urea, okay? So I'm pretty sure what happens here is because it's isoosmolar, this is the example that Subkan went through. You've got the table up before, so it becomes 600 because the urea is actually permeable. So it goes inside the cell and the osmolarity changes, which causes water to rush in and causes hemolysis. So those are your three types of questions which I have. I think there's one more. So there's one which says that, actually, I'll give you access to these questions. They're from another PSP that I had last year. So I'll give you access to the other ones because they're ones which you have to think about a bit more. But just know the ones which I explained because they're the most important ones.
All right.
All right. And last thing about effective osmoles, that's basically just another way of saying non-diffusibles.
Yeah.
Pretty much. Just as simple as I can put it is that, yeah. That's all I've got.
Yeah. Effective osmosis refers to basically what's like the tonicity as well.
Yeah.
Yep. Okay. So if we're done with that, I guess just some small housekeeping things. So, Khan? Yes? No? Okay. She's gone.
Hello.
Nope, she's back. Okay.
Hi, I'm back. What's going on?
Any housekeeping things?
I don't think so. We're trying to figure out how to do revision sessions on Zoom, right? So we'll keep you guys updated on how we think we're going to do that. And hopefully there'll be another one of these content sessions next week covering some biochemistry that I think you guys are covering in lectures this week.
In terms of biochemistry, basically me and my friend Ben will be going through that, and it will be pretty comprehensive. But if you were in Bio 3-4, we'll also be going over Bio 3-4 stuff, which you have to know for medicine. So don't be too concerned from that point of view.
Yeah. Just so on the revision things, more on that. So what happens is that we're planning on doing bi-weekly revision type things. So what we have got someone who made questions. So Anita's made some lovely questions for you guys all, so that they are designed to help you revise the concepts and be able to explain the concepts in your own terms, so that you can understand and make sure that you know what you're doing. And what we wanted to do was try and run sessions bi-weekly. So once every 2 weeks, and we actually wanted to, while we could go to uni, that everyone could come in and we would supervise you guys doing these questions. Unfortunately, that's not an option anymore. So what we will be doing is we will definitely be releasing these questions. They're on the drive. They'll be on the drives by next week for sure. So we'll see what we do with the sessions and whether we can do that with Zoom or whether we'll just leave you guys to your devices on those.
Last thing is, please friend us on Facebook. Please become our friend. Don't be worried about asking us stuff. Like, we were in the same position as you a year ago, and we know how daunting it can be. So if we didn't explain anything well today, if there's stuff that you still need clarification on, just please friend us on Facebook and message us, because we'll answer. And we also want to help you guys, because it's also low-key advantageous to us just revising year 1 content, which we need to know. So yeah.
We've also got intense cabin fever, please.
Yeah, everyone is dying.
Yeah, give us some interaction. But yeah.
Anyway, thanks for sticking with us.
Thanks for coming to the session. I think
spread the word. Spread the word.
Week. Yeah. Don't be hesitant to drop us questions or whatnot. It's good for both of us. But yeah.
Yeah. So next week we've got Nikita and Ben will be presenting. So yeah, hopefully we'll see you then.
And then maybe genetics. I'm not sure. We'll have to.
Yeah.
Maybe genetics,
we're lining them up with your lectures. So
yeah,
we'll see how it goes.
Anyway.
All right. Thanks for coming, guys.
See ya.
Bye.